The title of my presentation is The Fabrication of Vapor Crosslink Hyaluronan Polyethylene Interpenetrating Polymer Network for Flexible Leaflet Heart Valve Replacements. It's a fairly long title, but I'll make sure to go through each term to explain why they're all very important. So I just want to give you a quick background on the research. Heart valve replacement is one of the most required surgeries for people with heart diseases, and it is expected that the number of annual required surgeries could be as high as 150,000 by 2050. And it's fairly invasive because you do need to have an open heart procedures, and because of that, the risk can be high, and where many people cannot withstand the entire process. This is especially true for elderly patients where the mortality rate can be as high as 12.5%. And it's actually worse in developing countries where cost and scarcity of resources is a much bigger issue too. So our lab at CSU, we are trying to design something called flexible heart valve leaflets by making them out of linear low density polyethylene films, or LLDPE. And we can use this for something called transcatheter aortic valve replacements. And it's a big advantage over traditional method because TAVR does not need a open heart procedure. So we know that it's much less invasive. Another advantage of this technique is that because we're using LLDPE, which is fairly flexible and tough, we know that it's very cheap to make because this is the same material that is used to make Ziploc bags or plastic bags you see in grocery stores. So the example of this medical device is shown by the image on the right. Here we have one of the leaflets that's made from LLDPE. It's clear, it's small, it's very transparent. The main shortcoming for a lot of these synthetic polymers, like LLDPE, is that they tend to cause unwanted blood clotting or thrombosis. The solution to this that we have come up with in our lab is by enhancing the LLDPE films with something called hyaluronan, or HA, by making an IPN out of the two polymers. HA is, we know that it's blood compatible and biocompatible because it is ubiquitous in the human body, and it is present in your blood vessels and in the heart valve leaflets, in natural heart valve leaflets. And study has shown that it can be used for cardiovascular applications. And the chemical structure shown on the right here is an example of one repeating unit of HA. It is a sugar molecule that comes in long chains. Now, to form an IPN between HA and LLDPE, what we usually do is we swell the polyethylene and then penetrate it with the hyaluronan, and then cross-link the hyaluronan with the cross-linkers such as toluene diisocyanate, as shown by the schematic at the bottom of the slide. Here, even though the HA and the LLDPE are not covalently bonded, that highly interwoven network of the two polymers allow for the creation of one strong material. So after creating our biomaterial, we decided to do many tests to ensure that this can be used for cardiovascular applications. We did something called cytocompatibility studies, or two different studies in this case. One by directly grow human cells with the biomaterial, and the other study is to analyze whether or not any chemical species being released by our biomaterial could cause any cytotoxic effect to the mammalian cells. And the results show that our material is not cytotoxic, and it does not cause the cells to die during the culturing process. We also did an enzymatic degradation study to ensure that the crosslink HA does not get degraded by enzymes that exist in blood. And the study did show that where the HA were durable in a solution containing human enzymes, and it is physiologically relevant. To ensure that the material can be used for cardiovascular application, we actually did two different blood studies. One where we put a droplet of blood on the surface to see whether or not the material will cause more clotting than untreated LLDPE. And the second study, we took whole blood human plasma and incubated our samples and to see whether or not any platelets would adhere and activate on the surface. In both studies, we have shown that HA enhancement process does reduce clotting and platelet adhesion and activation compared to untreated aerial DPE. So these are great results. We have shown that our material is blood compatible and biocompatible, but these are static in vitro studies. So in the future, we do want to perform in vitro dynamic studies and perhaps consider doing in vivo studies too, because it is the most representative and most relevant if we want to put these implants in actual humans. So all of these studies I've shown you, they could not be accomplished without the help of my advisors, Dr. James Prow and Dr. Popat, along with student helpers, as shown by the left column on the slides. The center column shows all of the labs that have helped me throughout the entire experiments, and funding is provided by the NIH. Thank you for your attendance.